to me, I still find it incredibly fascinating that cells and tissues emit light. That sentence in and of itself is pretty cool. And um, recently, we also have shown that brain activity in human living subjects, that they emit light emissions that seem to be tracking what their activity is. So based on what their brain activity is, we typically use MRIs or EEGs, electrical signals. But we showed that we can detect the light coming out of someone's skull. First of all, that was pretty cool, that was meaningful.
I didn't
And
even know that you could do that.
exactly. So that was pretty exciting, is that the light coming out of someone's head can be measured. The caveat is, I mean, you need to have a hyper dark room. So we have a Faraday cage that is double dark chamber. So it's a dark room inside of a dark room that is lined with photo curtains. So it's extremely dark in there, so that we can pick up these subtle signals. So that's exciting, is that we can pick up these light signals. And the other thing that I've learned that right now is, I went into this thinking that the light that we are measuring is correlated, very strongly correlated to brain activity. And it can be, but it has opened up so many other questions that is really exciting. Is if there is light being emitted into the brain or being emitted by brain tissue and cells, how is it getting around? And if it's involved in meaningful information and processing, how are cells processing it? So right now we understand molecules and neurotransmitters, when they're being released, there's a receptor, and there's a place for all of these molecules. What does that mean for light? Are they using the same molecular landscape? Are there other ways that this information is being processed? So it's just opened up so many more questions for us to answer. Is the light being emitted just a byproduct of metabolism by our mitochondria? Um, the working hypothesis that we're trying to test, amongst many other, is no. I don't think it is just a byproduct, because there's some evolutionary evidence that suggests not. For example, within deep brains, like within your basal ganglia, so deep brain structures, there are proteins in there that are light sensitive. And these are proteins that are outside of your visual perspective. So they're not in your eyes. They have nothing to do with vision. So it kind of begs the question, what are these proteins and structures doing there that are light sensitive? And it's probably because we're emitting light, and they probably are processing that light. So there's some evolutionary explanation for why the photon emission might be occurring. Um, so that's the first part. Now, can we harness it? That's quite exciting. And so, if we were to think really big picture and far out there, we've developed as a community some really cool technology like fiber optic cables, right? We can send telecommunication from Canada to Europe using these telecommunication cables, fiber optic cables, to process information. If we understand how the light is propagated in the brain, for example, we can kind of repurpose some of this technology to attach fiber optic cables to brain tissue to propagate some of this light.
That's
Sounds
just
far
so
out there. It absolutely does. Totally understand.
that sounds so scary.
I mean, we have implants right now to help with electrical conduction, right?
Hmm.
To help with, in epileptic conditions or in Parkinson's, right? To jolt the brain, to help synchronize it, or even the heart. It doesn't have to be the brain. Our
Why?
biology is pretty good at getting rid of things that's useless to us, that is energy
Hmm.
inefficient. So we do have vestigial organs, and like the appendix, things like this. But proteins within the brain, something that the brain is highly efficient in terms of energetics, I think evolutionarily it would have been weeded out. So it doesn't make sense that every generation or evolution that this is recapitulated unless it serves a purpose.
Hmm.
This is speculation, but what we do know, and not to discredit some of the work that's been done by other researchers in this area, is that there's a reason also why the biology seems to respond to applied light. For example, infrared therapy has become widely used now. A lot of it is in the aesthetic domain, for example, but even also in repair and therapeutics. After an injury, a muscle injury, a joint injury, red light therapy has been shown to work very well.
Hmm.
Some people will suggest that it's heat. You're applying heat, so you're changing that domain. You're putting an energy into the system, but those are all terms. It's still light. The light needs to interact with biology. So I think we still don't understand how. The people are looking for like a single molecule that's going to resonate very well, but my view of this has completely changed. And that used to be my view. I used to think, hey, we need to look for a protein that's light sensitive, and that protein is going to send a signal inside, and that's the typical way that we think of this. My view has completely changed because I've started to think of biology as not just a biomolecular soup. Where I think reframing our understanding as our biology as an energetic landscape, that our brain is functioning at a set point of energy, of every action potential is an energetic signal. Every heartbeat is an energetic signal that seems to be related to some sort of molecular landscape. So when we add energy into an energetic system, it's like adding more water to a container. You can kind of change the way that it interacts with the whole system. So this is all working hypothesis, but I think it's kind of helping us to reframe how magnetic fields, how would they work if there's no receptor? How would light work if there was no receptor? It kind of helps push us this idea forward.
Fair.
The last year and a half has been incredibly transformative in this space because people are finding parallels to what we are showing. And for example, in the psychedelic community, people who are trying to understand altered states of consciousness, or how do these psychedelic compounds change our brain activity, that seems to parallel what we're finding in terms of light emissions. There's subjective experience of increased light experiences in these altered states. And that kind of begs us to ask, are they experiencing more light, or are they perceiving it? Is that something that they're kind of thinking about in these states?
Is that
Part of this study, we've developed a helmet, a light helmet, high dense array helmet that can pick up signals all over the head now. We were only working with 4 sensors. Now we are working with 16. So we have increased our density. So hopefully we can see, and we can answer this question. When people have altered states, are they emitting more light? I would have thought yes, but based on some preliminary data, it's actually not. There's less emissions coming out of their head. So the next question is, where's the light going? My hypothesis is when people are engaging in visualization, they're seeing in their own brains, basically, if you want to say that, the light probably is being propagated. It's being used. It's being moved around. And so this hypothesis, something that I'm really passionate about, that we're working on actively right now, is we are very confident that light emission is not a metabolic byproduct, I think. The second point is, if it's not, how is it getting moved around? So we're doing this light propagation experiment where we're taking axons that are aligned, that typically propagate what we think electrical information. Can it basically be looked at as a fiber optic cable? The properties of when we look at these axons can act like a fiber optic cable. So we're doing some experiments to actually prove that. If they are, then we have really good foundations to say that the light that's being emitted can get moved around in these biological structures. If not, I'm not sure what this means.